Welcome to Chapter 17 of Anatomy and Physiology, and with this chapter we finally finish off the nervous system by talking about your special senses. You remember special senses way back from kindergarten. Those are things like vision, hearing, smelling, tasting, and something called equilibrium. That's all we're going to be talking about to finish off nervous system, because to help you to do all those sensations, remember sensory is a function of the nervous system. Well, it's going to be your nervous system that's really helping you with these special senses. So this is going to be the last part of this system, your special senses. And like always, we're just going through anatomy and physiology. We're going to look at the organs in your body that help you to see, smell, taste, and hear. And then we're going to talk about what all the different parts do, specifically to help you to do or sense those different stimuli we call your senses. So we're just going to go organ by organ, sense by sense, talking about how it works. And we're going to start off with the first sense called olfaction. Oh, you remember olfaction just means smelling. So we're talking about smelling. So you already know basic things. You already know you use your nose to smell. If you think back to cranial nerves, hopefully you remember it's going to be cranial nerve 1 helping you to send that sensory information. So you know some things already, but like usual, we're going to go in a little more detail. And we're going to start off by looking at the microscopic anatomy. What structures microscopically are helping us to smell? Well, it'll help if I give you a hint. Turns out smelling, just like taste sensation, is really a chemical sense. When you smell something, when you have an odor, what you're really doing is sensing a chemical, and your brain is interpreting that as a smell. So smell is really a chemical sense. And you're going to need a cell that helps to sense these chemicals in your nasal cavity. These cells are going to be part of your nasal epithelium, specifically helping you to smell. So we're calling them part of your olfactory epithelium. So if you were to look in the nasal cavity, kind of on the roof of the nasal cavity, you would find some olfactory epithelium. And that olfactory epithelium will have cells that will go through the tiny holes in your ethmoid bone. Think back to bone. Remember, those were your cribriform plate that had the tiny holes. They will go through the tiny holes of the cribriform plate and go on up and synapse or communicate with neurons forming part of your olfactory nerve. So when you think about it, when you take that road trip with your friend and your friend farts in the car, what came out of your friend's butt is now touching a neuron that's associated with your nervous system. Oh boy. And that's what you're seeing in this picture. You're seeing a picture of the nasal cavity, and they're trying to zoom in on that little box showing your ethmoid bone's cribriform plate. You're seeing the yellow nerve, olfactory nerve sitting on top of the ethmoid bone, and that purplish area is where you're going to have a lot of those chemicals receptor cells, those olfactory epithelial cells. So it's just your body sensing chemicals. And so who's really doing the sensing? Who's doing the work? Oh, we got to talk about the cells. Turns out when we look in the olfactory epithelium, we find 3 major groups of cells. I want you to know these 3 major cells, and basically what are they doing? What is their function when it comes to smelling? There's the olfactory receptor cell, the support cell, and the basal cell. So what are those 3 cells doing? Well, like always, they give you a hint in the name. Like the first one, it's the olfactory receptor cell. Well, this one is telling you olfactory for smelling, receptor because they're sensing the chemical stimuli. These are the cells that are responsible for smelling. They notice the odor. They sense the chemical. And then there's the support cell. Well, it tells you in the name. Support cells are simply providing support. How? They could provide nourishment as well as structural or physical support for the other cells. And then there's the basal cell. Oh, we've seen basal cells by now. Remember, basal cells, basal layer meaning bottom layer, these are cells that typically are stem cells. They're typically cells that replace other cells. That's what these are. So your basal cells can replace other olfactory receptor cells when those cells get old and they die. So those are the three major cells that are helping you with your olfaction, that are in the olfactory epithelium. And that's what you're seeing in this picture. Over to the left, from top to bottom, you're seeing the big portion of the olfactory nerve, that big round portion at the end of it called the olfactory bulb. It's sitting on top of the cribriform plate of the ethmoid bone, and you see all the tiny little foramina. And then you see the cells sending their axons through the cribriform plate. You're seeing the olfactory epithelial cells, or the olfactory receptor cells. They're the ones in blue sending their long blue axons in the cartoon. And then in between them, you see these large, long cells not sending an axon to the nerve. Those are the support cells, providing nourishment, protection, and even just structural support. And then you see these tiny, tiny cells kind of clustered on one end. Those are the basal cells. They're just immature stem cells waiting to replace other cells when the other cells die. And so that's really what's in your olfactory epithelium. And again, make sure you know what those cells are doing. And again, here it is on this slide. Those support cells, they're just supplying support. What type? Physical support, nourishment. They can even provide insulation, very similar to how myelin insulates an axon. Well, these support cells can insulate these olfactory receptor cells to help enhance the movement of their electrical activity. Oh, yeah. Then those basal cells, I told you before, they're just stem cells. So they'll replace other cells, specifically those olfactory receptor cells, so that you could keep on smelling. And we already know the olfactory receptor cells are responsible for sending that chemical signal, that smell signal, that olfaction signal. And like always, there's other things there. When you look in the nasal cavity, there is other things in that olfactory epithelium. Let me give you a hint. We're talking about the nose. What else is in your nose sometimes? Oh, it's boogers. What's boogers? Well, it's really a lot of dried mucus. And so where does mucus come from? Well, mucus comes from glands. Turns out you have olfactory glands, sometimes called Bowman's glands, that are producing a mucus. And that mucus is there to actually help dissolve that chemical. Remember, the odors are chemical. So when that chemical enters the nasal cavity, it will touch the mucus made by these olfactory glands, and it will dissolve, and it will be easier to sense and transmit that signal using your olfactory receptor cells. And we even give a name to this. Remember, in anatomy, we name everything. Turns out you're taking an originally chemical signal, that odor was a chemical, and you're going to turn it into an electrical signal. Remember, your neurons are going to communicate by making action potentials. We give a name to this. When you turn something like a chemical or a molecule signal into an electrical signal, we call that transduction. So in order for you to smell, you need to do transduction. You need to turn that molecule, that chemical, into an electrical impulse. Why? Because that's the way your neurons are sending information in your brain. They're zapping each other with action potentials. Don't forget nervous tissue. And so we can basically track the pathway of information for smell. I want you to know the pathway of information for smelling, or olfaction, and that's what you're seeing on this slide. So again, let's say you're taking your road trip with your friend across the country, and they fart in the car. That chemical left your friend's bud. It entered your nasal cavity. Then what? Well, it's going to touch one of those olfactory receptor cells in your olfactory epithelium lining your nasal cavity. And then it's going to send a signal using the axons, which are going to pass through the foramen in the cribriform plate. And then after that, it will synapse with neurons in the olfactory bulb, like I mentioned before. Remember, olfactory bulb was just that rounded end to the olfactory nerve. Remember, just look at your cranial nerves pictures. And then after that roundish end to the olfactory nerve, is a long strand-like extension. We call that the olfactory tract. And that olfactory tract will send that information to the brain, to a specific part of the brain responsible for processing this olfaction information. It's called the primary olfactory area, which happens to be in your temporal lobe. So it turns out we're going to see temporal lobe multiple times. We're going to see it for smelling, because the primary olfactory area is in the temporal lobe, and we'll also see it later when we get to hearing. But again, know the pathway for smell. Hit olfactory receptor cells in the olfactory epithelium of the nasal cavity. Send the information through the holes in the cribriform plate using axons, which will then synapse with the neurons in the olfactory bulb, which will then send it on to the olfactory tract, which will then send it on to the primary olfactory area of the temporal lobe. And that's what you're seeing in this picture. Starting off in the left, that zoomed in picture of the nasal epithelium again, you see the chemical odors, or the little dots at the bottom. You're going to hit the olfactory receptor cells. They're sending the information through the foramina and the cribriform plate. Then they're going to synapse, or come into contact with, communicate with neurons in the olfactory bulb, which will then send it on to the olfactory tract. Then over to the right, you see the rest of it. After the olfactory tract, it's making its way to the olfactory area of the temporal lobe. And that's the pathway of information for smelling. And I told you it all began with you first taking this initial chemical signal and turning it into an electrical signal. All you have to do, transduction, specifically for smelling, specifically for olfaction. So we call it olfactory transduction. And this happens as soon as that chemical binds to your olfactory receptor cell, specifically to a protein on the olfactory receptor cell called the olfactory receptor protein. And once that chemical binds to the olfactory receptor protein, it's just going to trigger a sequence of events that will lead to the formation of an action potential. And it turns out you know some of this already, because you know how to make an action potential. You know one of the first steps involved in making an action potential involves depolarization. So we know when that chemical binds to that olfactory receptor protein, depolarization is going to occur. How? Well, I told you there's going to be a sequence of chemical reactions. You do not need to know them. But these sequence of chemical reactions are going to involve a special compound called cyclic AMP, sometimes abbreviated lowercase c AMP. This chemical is what's going to help cause the depolarization in the neuron that's going to lead to the formation of an action potential. And then that action potential is going to make that long journey to the primary olfactory area of the temporal lobe. And you'll identify that there is an odor, or you'll identify at least that there is a signal. But you won't be able to identify the odor until you send it on to one more place. So actually, after the primary olfactory area of the temporal lobe, you're going to send it one last place. You're going to send it to the frontal lobe. Oh, why? You're sending it specifically to the orbital frontal area of the frontal lobe. This is where you're going to name the odor. So again, one more time, if your friend farts in the car, again, that chemical is going to bind to a receptor in your olfactory epithelium, which will then pass through the foramen in your cribriform plate, synapsing with neurons on the olfactory bulb, which will then go to the olfactory tract, which will then go on to the primary olfactory area of the temporal lobe, where you'll know that there is a smell. But then you won't be able to say, ew, it's stinky. You won't be able to identify the smell until you send it on to that last place, the frontal lobe, specifically the orbital frontal area of the frontal lobe. That's when you decide what that chemical odor is. So that's a little bit on smelling. And this picture is just showing you some of the chemical reactions that's going to lead to depolarization. And you know what depolarization is. You know that's the cell becoming positive on the inside because you've opened up voltage-gated sodium channels and allowed sodium to rush in. So you're seeing that last part over to the right. But over to the left, you're seeing the beginning. You're seeing that olfactory receptor protein. And what's going to happen when you smell is an odor, a chemical is going to bind to it and trigger a sequence of events that will lead to the formation of cyclic AMP. And it's cyclic AMP that's going to act on these sodium channels to allow them to open and allow for depolarization. But again, stick to your exam review. And that's it. Now you know how to smell. It was that simple. We went through the structures, looked at who's it responsible for doing smelling, and then we talked about how they did it. That's all we're doing for your special senses, breaking down the anatomy and then talking about the physiology, like always. So we're going to do the same thing for this next sensation, for this next special sense. It's for taste. And like always in anatomy, we have a special name for it. The sense of tasting is called gustation. To gustate is to taste. And I'd mentioned earlier, tasting, just like smelling, is a chemical sensation. And luckily for us, it's a little simpler than olfaction. And like always, you know some things already. You know, when it comes to tasting, we could identify different flavors or primary tastes. There are usually 5 major primary tastes. There's something called sour, of course, sweet, bitter, salty, and then there's this last one called umami. Oh, what's umami? It's usually described as a meaty or savory flavor. Kind of think Thanksgiving, when you're having your Thanksgiving meal, that tends to be very savory. You're going to taste a lot of umami flavor. But again, we got to go through the anatomy and the physiology. Who or what is helping us to taste and how is it happening? Well, first up is the anatomy. What's involved? Well, it's kind of like smelling. It's going to be cells again. Remember, this is really nervous system. It's just a whole bunch of neurons sending info. So what cells and what do we call the cells and where do we find the cells responsible for taste? Well, again, you know something. It's going to be on the tongue. Remember, you could taste on the tongue using cranial nerve 7 on the anterior 2/3. You remember you could taste in the back of the tongue, posterior 1/3, using cranial nerve 9. And you could taste in the throat, in the pharynx. So those are all places where we're going to find these cells responsible for allowing us to smell. And these cells are going to be in special structures we call taste buds. So you tend to find taste buds on all the places you could taste. You find them on the tongue, your throat, and even in your epiglottis. You'll have taste buds. And if you were to dissect the taste bud, cut it open, we're going to find cells there. And we're usually going to find 3 unique types of cells. Oh, well, again, I want you to know now these cells and what are they doing to help you to taste. And they're going to sound a lot like the olfactory epithelial cells. What are the cells now for taste? Where there is the gustatory receptor cell. Again, I'm seeing support cells. And again, I'm seeing basal cells. You might know what these 3 cells do already because they're telling you in the name. Again, to taste is gustation. So the gustatory receptor cell, well, that's going to help you to taste. Then there's support cells. It's the same type of cell as the support cells for olfaction. These cells are just providing basic support. What type? Nourishment, protection, structural support. Again, also insulation. Now for the gustatory receptor cells. And again, we have basal cells, which are still stem cells to help replace the other cells when the other cells die. Those are the same 3 basic groups of cells. You have the receptor to notice the chemical, you have the support cell to provide support, and you have the basal cell to replace everybody when they get old and die. And again, there's images, and that's what you're seeing in this picture. Off to the left, you're seeing the surface of the tongue, and on the surface of the tongue are just small little finger-like projections we call papillae. But you'll also see taste buds. And if you were to zoom in on a taste bud, you'll see an image like what you see over to the right. You're just seeing a cluster of cells. And again, it's going to be the same three major groups of cells. There's the cell to sense. It's the gustatory receptor cell, sensing the chemical, which you would identify as a flavor, a taste. There's going to be the support cells, providing structural nourishment support. And towards the bottom, you see the basal cells that will replace other cells. And you see, again, in the cartoon, they made the receptor cells blue, and you could see its axons of the neurons going on to synapse with the nerve when they're ready to send the information. But again, more anatomy to go over. I mentioned that you find these cells, the gustatory receptor cells, support cells, and basal cells, in taste buds. And we saw in the previous picture, you can find taste buds on the surface of the tongue with other finger-like projections I called papilla. Turns out on the surface of your tongue, you have a lot of these finger-like projections. You have a lot of papilla. So like always, we're going to classify them. I'm talking about all these little bumps you see on the surface of your tongue. If you were to flatten out a piece of gum on your tongue and pull it off, peel it off, it would look like there were little pins sticking into the gum. You were seeing all the little papilla pushing into the gum. It's all those little bumps on the surface of your tongue. And there's a lot of them. So we classify them. There's major groups. There's what we call the valate papilla, the fungiform papilla, the foliate papilla, and the filiform papilla. What do I want you to know? They are all papilla. They are all finger-like projections. I want you to know a difference. Turns out, most of your papilla have taste buds, but not all of them. I want you to know which one of your papilla, which one of those finger-like projections, which one of those bumps on the surface of your tongue do not have taste buds. Turns out of all four of the valate, just in case you're using another textbook, they'll sometimes call the valate circumvallate, the fungiform, foliate, and filiform. Which of those four does not have taste buds? Turns out it's the filiform papilla. The filiform papilla does not have taste buds. And unlike the other papilla, if you go back to the previous slide, the other papilla, you tend to find them in certain places on the tongue. While the filiform papilla, you find everywhere on the tongue. Why? And it turns out if you were to look at them and compare them, they don't all look the same. Turns out the other three, the valate, fungiform, and foliate papilla, they tend to look a little bit rounder and they'll have taste buds. But if you were to look at a filiform, it will look more sharp and pointy. And again, you will not find taste buds. Why? Why does it look sharp and pointy if you were to look at it? It has to deal with the function of this one. The other papilla, because they have taste buds, they are going to be involved in tasting. But the filiform is actually there almost as a tool. It's there to help increase friction, to make it easier for you to grip and move food with your tongue. That's what's happening. That's why when you kind of flatten out your gum on your tongue, it doesn't just slide off your tongue because your tongue is gripping that gum with some filiform papilla. So keep it simple. Know your valate, foliate, and fungiform papilla have taste buds, and your filiform do not. Why? Because your filiform are there to increase friction and make it easier for you to grip and move food with your tongue. And again, pictures. That's all you're seeing in this picture here is just the surface of the tongue, and they're pointing out the different papilla. The easiest ones to identify are the valate. They are in a unique place, and they have a unique orientation. The valate are towards the back of your tongue, and they tend to form a little V at the very back of the tongue, and they're large papilla compared to the other papilla on your tongue. So if you were to go in the bathroom and say, you'll have to stick your tongue out as far as you can and look way in the back, you'll see these large round circles forming a V. That's your valate papilla. They are still there to help you to taste. But again, we have to follow the trail. We know the trail for information, for smelling. Well, we're going to have to do the same thing for taste. I want you to know the pathway of information for taste. And again, you know the beginning from our previous slides. It's going to begin with, of course, you tasting something, putting it on your tongue, for example, and having it get picked up or sensed by gustatory receptor cells. And those gustatory receptor cells are going to send that information on somewhere else. Well, where are they going to send the information? Well, it depends on where that cell is located. You know this from cranial nerves. If that gustatory receptor cell is on the tongue, the anterior 2/3 of the tongue, well, you'll send the information using facial nerve, aka cranial nerve 7. But if that gustatory receptor cell is on the back of the tongue, the posterior 1/3 of the tongue, you're going to use the glossopharyngeal nerve, aka cranial nerve 9. Or if that gustatory receptor cell is in the throat or the epiglottis, well, you're going to use cranial nerve 10 or your vagus nerve. Remember, this is VAGUS, vagus, not Las Vegas, Vegas. But that's where you're going to send the information. So it depends on where you're sending the info from. You're going to use one of these 3 nerves. And then where would you send the information next? Again, lots of pictures. That's what you're seeing here. Again, you're seeing information coming from those different areas, the front, the back of the tongue, and the epiglottis and throat areas. And then they're going to have to send the information on using those nerves. Turns out you're going to go through the medulla oblongata. Turns out you're going to go to a collection of neuron cell bodies in the medulla oblongata called the gustatory nucleus. Well, it tells you the name. It's involved with tasting. It's going to help to process some of that tasting information before it sends it on up. And this is a type of sensory information. Oh, you remember a lot of your sensory information needs to be fine-tuned before you kick it on up. Oh, you remember where do you send information, sensory information, except for smelling? Well, it's the thalamus. This sensory information, this taste information has to make a stop at the thalamus before it gets sent one more place, before it gets sent to the primary gustatory area. So that's really the pathway for tasting. You're going to start off again with the gustatory receptor cells, either on the tongue, front of the tongue, back of the tongue, epiglottis, or throat. Then you're going to send that information on using one of your 3 cranial nerves, either cranial nerve 7, 9, or 10. Then you'll send it on to the gustatory nucleus of the medulla oblongata, then the thalamus, and then you'll end at the primary gustatory area. That's the pathway for taste. And now you know about tasting. Oh, you got 2 senses now. But again, lots of other senses to go. Here's another one. We're moving on to vision. Now, it's to see. Again, like usual, we're going to do the same thing. We're going to talk about what it means to see. We're going to talk about the structures that help you to see. And we're going to talk about how do you do it. That's all we're doing. And now we're moving on to vision. How do you see light? Well, when we talk about seeing light, we're really seeing what's called the visible light. And the visible light is only a small portion of all the light that exists. And we tend to map out or arrange all the different lights that exists in terms of their wavelengths. And we call this spectrum of wavelengths for light the electromagnetic spectrum. And it includes a lot of things you might not consider light, like gamma rays or X-rays, and yes, even microwaves. The waves in your microwave are microwaves. They're actually types of light. And what we see is visible light. And it's only a small portion of all the light that exists. And we can measure these different lights based off their wavelengths. And the light that we see, that visible light, ranges from about 400 nanometers to about 700 nanometers when you look at their wavelengths. And by the way, just in case you don't know, what is a wavelength? It's just the distance in peaks between consecutive electromagnetic waves. These types of light, like all light, travel in waves. And we measure the peaks of adjacent waves, and we call that a wavelength. No, you don't need to know about wavelengths. Remember, just stick to the exam review. But back to vision. We got to talk about vision. Remember, we're just going to break down the anatomy, what's helping you to see, and talk about the physiology, how they're going to help you to do it. And like always, you know some basic structures. We're talking about the eye and things in and around the eye. And especially when it comes to things around the eye on your face, you know what's there. Well, it's going to be things like your eyelids. Well, they're there partially for protection. There's going to be things like your eyelashes and your eyebrows. Turns out they're also providing protection. You will have your tear-making system. We call it the lacrimal apparatus. And you're going to have things like muscles to help you to move the eye. So we're just going to talk about all these basic structures around the eye, and then we're going to cut open and look inside the eye. So what do we see when we look around the eye? What's some external anatomy? Well, there's going to be muscles. For example, there's the muscles in your eyelids that help you to open your eye. Remember, your orbicularis oculi only closes the eye. Well, turns out to open the eye is muscles in your eyelids. We call them your palpebral muscles. Palpebral muscles are muscles in your eyelids that help you to open the eye. There's even muscle that attaches to the eye from the outside. We call it your extrinsic eye muscles. And it's going to be these extrinsic eye muscles, these muscles that attach to the eye from the exterior, are going to be involved with moving the eyeball. So when you move your eyeball, there's no muscle inside of the eyeball itself moving it. It's actually muscle attached to the eye from the outside. And then it's like your eyeball also has a little vacuum seal covering. It has a little layer of mucous membrane around it we call the conjunctiva. It is a clear membrane around the eye that lines not only the eye, it also lines the inside of the eyelids. It's clear, it's translucent. It's what that shiny look is to your eye and the inside of your eyelids. You're just seeing some conjunctiva. And this could sometimes get irritated, by the way. And when it gets irritated, when it gets inflamed, blood vessels here will kind of dilate and it will look pink. You can call it pink eye. Pink eye is really called conjunctivitis. It's just when you get inflammation of the conjunctiva. You've inflamed or irritated it. How? Usually due to some type of infection. Pink eye. But remember, if you know anatomy, it just helps to explain stuff. Now you know when you get pink eye, you're just irritating and inflaming this conjunctiva, usually because you got an infection. But keep going, there's other stuff here. And you remember in anatomy, we name everything. We name lots of weird things. For example, there's a fold of connective tissue that forms the eyelids. It's called the tarsal plate. Tarsal plate is what's helping to form the eyelid. So kind of think that sheet of tissue that folds down in addition to the palpebral muscle and the conjunctiva lining the inside. It's mainly this connective tissue we call the tarsal plate. And this tarsal plate is kind of weird. It actually has some sebaceous glands in it. These are sebaceous glands, so they're making sebum. Why? Turns out it's lined up in a row. It's a row of sebaceous glands we call the tarsal glands because it's in the tarsal plate, the eyelids. And it's keeping the eyelids from sticking together. So your eyelids make a little oil that keeps them from sticking together. Oh, yeah. So if you ever wondered, why do my eyelids feel a little greasy? Well, you're just seeing a little sebum. And again, lots of pictures to help show a lot of these structures. Again, you know where a lot of things are. Superior to the eye are the eyebrows. Anterior to the eye is the eyelid with the eyelashes. And in this cartoon, you can see a cross section into the eyelid, or a longitudinal section into the eyelid. And you're seeing there's some muscle inside. That's some of that palpebral muscle. And you're also seeing in green the tarsal glands. You're seeing where there's going to be sebaceous glands making sebum to help keep the eyelids from sticking. But again, lots of other structures to see in the eye. So we got to keep going. What else is there for the eye? Well, I told you there is the tear-producing system. There's the system or apparatus structures in the eye that help to form and drain tears. It's called your lacrimal apparatus. I want you to know the pathway for tears? What is the lacrimal apparatus? What are the structures that are helping to make and drain the tears? Well, you're seeing the full pathway here listed out. Simply, it's first you're going to make the tears in the lacrimal gland. Your lacrimal gland sits more superior and lateral to the eye. So kind of think superior and lateral, kind of behind the sides of the eyebrow. You'll have your lacrimal glands. And you'll need to drain them. They just make the tears. You're going to have to drain the tears out of the glands. Well, you're going to drain it using a duct called the lacrimal duct. And the lacrimal duct will drain the tears over the eye, and then you need to drain the tears. You can either cry the tears out, or you could drain them. Turns out, if you were to look in the eyelid, you'll see a tiny hole. We call that the lacrimal puncta. That's where you'll drain the tears. Little kids are really good at using it. If you ever seen a little kid about to drop a tear, and you're like, don't cry, don't cry. You want some candy? And that tear vanishes. And you're like, where did the tears go? They drained it into their lacrimal puncta. All kids are great at it. And then after the lacrimal puncta, you'll drain it through a larger canal called the canaliculi, which will drain into a large dilated sac called the lacrimal sac. And it will drain out of the nasal cavity using the nasal lacrimal duct. This helps to explain stuff. You can see the end to the tear pathway ends in the nasal cavity. If you ever wondered, why is it that my nose always runs when I cry? Now you know. Yes, there are some boogers in there, but you're also having tears. You cry out of your eyeballs and your nose. Ew. But that's anatomy. And like always, there's pictures, and that's what you're seeing. Let's track that flow again using the pictures. So in this picture, again, kind of superior and lateral to the eye, you see this huge green cluster in the cartoon. That's the lacrimal gland. That's where you'll make the tears. Then you'll need to drain it. So you see these little tubes draining or leaving the lacrimal gland and going towards the eye. That's the lacrimal ducts. Then the tears will drain or wash over the eye. And yes, you could just cry it out. Or you could drain it through these little small pinpoint holes in the eyelids. Those are your lacrimal puncta. And then you see these tubes leaving the eye. Those are your lacrimal canaliculi, plural. Canaliculus is singular. Then as you approach the nasal cavity, you'll dump into this large dilated sac. That's the lacrimal sac. And then you'll use that long tube to drain through the nasal cavity. It's called the nasal lacrimal duct. So that's the order of flow for your tears. And your tears can help provide protection. If something gets in your eye, your tears are just going to be there to flush it out. It's part of a protection mechanism. It's part of your immune system. But those were just structures helping the eye, protecting the eye. What about the eye itself? Once we peel back the conjunctiva, peel back the muscles on the eyelid and on the outside of the eye, what are we going to see? Well, actually, I didn't really mention the muscles on the outside of the eye. Before we get into the eye, we got to talk about the muscles that help to move the eye. There are lots of muscles. We're just going to mention some. And a lot of their names sound a little weird, but they're really helping you out. So we're talking about extrinsic eye muscles. They tell you in the name, extrinsic means outside. They're attaching to the eye from the outside, and they're going to be responsible for moving the eyeball. There's something called the superior rectus, inferior rectus, lateral rectus, medial rectus, superior oblique, and inferior oblique. Their names tell you multiple things. Their names tell you where to find them, and in most cases, what direction they move the eyeball. So, for example, the superior rectus, if you were to look at a picture of the eye, which we will do in a second, the superior rectus is going to be up top. It's superior. And it happens to pull the eyeball in a superior direction. It gets you to look up. So you find the superior rectus superiorly, and it gets you to look up superiorly. While the inferior rectus, well, you can figure that one out then. The inferior rectus is going to be on the bottom of the eye, and it's going to get you to look down. The lateral rectus is going to be on the sides of the eye. Think ear side. Is lateral side in the face. So it's going to be lateral side, and it'll get you to point to the side, look to the side. And then your medial rectus is located medially. It's located nose side of the eye, and it's going to let you look medially. Kind of think looking at your nose. And then there's the last two. There's the superior oblique and inferior oblique. Superior oblique is located to the top and off to the side. It's all off obliquely. And inferior oblique is located down and it's off to the side. But their direction they cause you to look is kind of the opposite. How? The superior oblique gets you to look down and to the side. Oh, yeah. So when you look down to the bottom right with your right eye, you're using your right superior oblique. It gets you to look down and to the side. While the inferior oblique gets you to look up and to the side. So those are your extrinsic eye muscles and where they're telling the eye to move. And then, again, once we peel off those muscles, we could finally look at the eye itself. And your eye is a big round ball, and it's pretty hollow on the inside. It's actually filled with fluid. It's not a completely solid structure. And it actually has layers to it. Your eye has really 3 major layers. I know your textbook breaks it up into 2 tunics, but one of those tunics I actually separate and call it 3 layers. So what is it? There's something called the fibrous tunic, the vascular tunic, and your lab manual also mentions something called the sensory tunic. What's going on? Well, the fibrous tunic is really the outside of the eye. And the outside of the eye is really what you see when you look at the eye from the outside. It usually looks white when you look at the eye from the outside. That whitish outer part of your eye is called the sclera. And it's everywhere except for the very front of the eye. In the front of the eye, it's not completely white. How do you know? Because you can see people's eye colors. Some people have brown eyes, blue eyes, hazel eyes. Marge Simpson has hazel eyes. I don't know if you know that. How do you know? Because the sclera is not there. That's how you could see the eye color. If the sclera was there, you would just see a completely white eyeball. So instead of having sclera in the front of the eye, it's actually a clear coat in the front we call the cornea. So the cornea in the front and the rest is the sclera. That is the most superficial part of the eyeball itself, and we call that the fibrous tunic. If you were to peel that back, we would see a layer of blood vessels, and it's a structure we call the choroid. It's a layer of blood vessels. We could also call it the vascular tunic. And then there's an innermost layer, we'll see it later, that's actually responsible for sensing the light. It's called the retina. That's sometimes referred to as the sensory tunic. So your eyeball actually just has layers to it. And again, here's that picture of the eye, and we could see some of these structures again. So we already looked at things like the eyelid and the eyebrows and eyelashes and the tarsal glands. Look at the eye itself. So you can see the eye in the front. They're trying to label these structures. You see in the front, it's a clear coat. It's called the cornea. While the rest of the outside is that whitish outer layer called the sclera. That's all the fibrous tunic. Then you see a little tail light coming out the back of the eyeball. That's the optic nerve. Oh, remember that's cranial nerve 2, responsible for vision. It connects right into the eyeball. And if you look closely at the cartoon, if you follow that optic nerve, it's yellow in the cartoon. It kind of splits and becomes an innermost layer in the eye. That is some retina. So your eye has multiple layers, and there is a choroid, but this picture doesn't really do a good job of showing choroid. This picture does a better job of showing all those layers. You're seeing a big eyeball in this picture. And again, towards the front, it's slightly blue in the cartoon, but it's completely clear in your body. That's the cornea. Then the rest of the outer layers, the white, they're pointing to it off to the right side. That's the sclera. Then there's a middle layer, and you see little blue and red dots representing blood vessels, arteries, and veins. That's the blood vessel layer, the vascular tunic. That's the choroid. And that innermost layer of yellow is going to be your retina, sensing the light. And it's going to send that information on to the optic nerve. So we got to talk about that. How do I get that information to my optic nerve and all the way eventually to my occipital lobe. Well, what am I getting? Well, I'm trying to sense light. How do you let light into the eyeball? Because that sclera, even though it's white, it doesn't do a good job of letting light in. There's a reason why the front of the eye has that clear cornea. It's to let light in. And if you were to actually look into the cornea or look behind it, your eye actually has a hole in it behind the cornea. And we name the hole. It's called the pupil. And that hole is really thanks to some muscle, a circular ring of muscle we call the iris. You know your iris more as the colorful part of your eye. When you talk about having brown eyes, blue eyes, hazel eyes, green eyes, you're really talking about the color of the iris. And your iris actually allows you to change the size of your pupil. Your pupil, that little hole in the front of the eye, could get small, pinpoint, constricted, or you could dilate it, make it big. And you're doing it thanks to the iris. Why? Why does the iris sometimes make the pupil tiny or big? It's about light. Turns out when you're in bright light, think if you go outside during summertime with all that bright light, your pupils are going to shrink. They're going to contract to let less light in so you don't damage your sensitive retina. You don't want to overstimulate it with too much light. That could blind you. So you'll constrict the pupils a bit. But let's say it's nighttime and you're bouncing around, you're stumbling around in the dark. Well, to help you to see a little better, you're going to dilate the pupils to let in more light, to let it be a little easier for you to see. That's why your pupils change size. It's to help adjust to light. That's why sometimes when you go to the eye doctor, the ophthalmologist, they sometimes have to dilate your pupils. And when you leave, you got to wear that weird little flimsy sunglasses. They're trying to protect your eyes because that medicine they gave you is inhibiting constriction of the pupil. So your normal ability to inhibit light from coming in won't happen and you might damage your eye. So you got to wear those stupid little glasses whenever you leave the doctor and they dilate your pupils. Why? Because science. Now you know, anatomy. But anywho, so how's the pathway of light gonna go? Well, you're gonna allow light to come into the eye through that clear cornea. Then it's gonna pass through the pupil. And it's going to pass through the inner portions of the eye until it hits the retina, the innermost layer. So it's kind of weird. That light has to enter the eyeball and hit the innermost layer. It has to hit the retina. And then after the retina, we saw from the cartoon, you're going to send that information on using your optic nerve. So your optic nerve actually comes out the back of the eyeball. So there's a part of your eye where there's no retina. It's only optic nerve. Turns out it's actually a hole in the back of the eye for the optic nerve as well as blood vessels. And there's no retina there because it's a hole. So you're not going to be able to sense light where that hole is in the back of the eye. You're going to notice that as your blind spot. Your blind spot is thanks to a hole in the back of the eye to allow the optic nerve and blood vessels to enter and leave. We call it the optic disc. So there are some exercises online. If you wanted to find your own blind spot, you possibly could be able to do it. And it will just look like a black dot. And you're not able to see anything in that black dot because that's your blind spot. It's because where you don't have retina, because that's where a hole is to allow the optic nerve and blood vessels to enter and leave the eyeball. And if I were to look at the inside of the eye, you could see the retina. When you go to the eye doctor and they look inside your eye, they are using what's called an ophthalmoscope. Your ophthalmologist uses an ophthalmoscope to look at the retina. And when you look at the retina, there's going to be a part of the retina where you're going to have a lot of the sensory cells for light. We'll see these receptor cells a little later. And it's going to be where you have a lot of them. So it's going to be where you have your best vision. Your best vision is what we call your highest visual acuity. And we name this area in the retina where you have your best vision, where you have your highest visual acuity. It's called the fovea centralis. And the fovea centralis is in the middle of the exact center of the retina. Remember, we name everything. The middle of your retina is called your macula lutea, and in the middle of that, you have your best vision in an area called the fovea centralis. Let me show you in a picture. Here's a picture showing the retina. It looks kind of orangey red, and you're seeing blood vessels are the little creepy red lines. And if you look closely, it looks like all those creepy red lines, those blood vessels, seem to be originating from that little bright spot. What you're seeing is the hole in the back of the eye. You're seeing the optic disc, and you're seeing the blood vessels enter the eye from that optic disc. But over to the right, you're seeing this dark little red spot. It looks dark because there are a lot of cells there, and it's blocking light. So that dark area is where you have a lot of your receptor cells for vision. That's where you're going to have your best vision. That's your fovea centralis. And it happens to be located in the middle of the retina, which is what we call the macula lutea. So that's all we're talking about, just pointing out anatomy. And I told you in that fovea centralis are where you have the cells sensing light. It's where you have your receptor cells. And they're sensing light. We call them photoreceptors. And we actually give a name to the different types of photoreceptors, the different cells sensing light. There's rods and there's cones. I want you to know the different types of light they let you see. And it's on the slide. I'm going to keep it just as simple as the slide. Turns out rods help you to see dim, sometimes called black and white light, while cones help you to see bright, colorful light. So if you were to look at the retina, it's kind of like olfactory epithelium, where you're going to see different groups of cells. So the photoreceptors are not the only cells there. They're just the cells responsible for sensing the light. So when we look at the retina, it actually has different layers to it. It has the layer where you're going to find the photoreceptors, the rods and cones, where the rods are sensing dim, black, white light, and the cones are sensing bright, colorful light. Then there are going to be different layers, and they're going to have to send that information through the different layers. And that's what you're seeing on this slide. Soon as that light goes through the cornea, goes through the pupil, and hits the rods and the cones, the photoreceptors in the retina, they're going to have to send that information through other layers of the retina before it gets to the optic nerve. They're going to have to pass through a layer called the outer synaptic layer. Then they're going to have to pass that information on to bipolar cells. Remember, these are just a type of neuron. Bipolar cells. And through the inner synaptic layer to ganglion cells, which then send that information on to the optic nerve, or aka cranial nerve 2. And again, pictures help. That's what you're seeing in this image here. So over to the left, you're seeing the eyeball, and they're zoomed in on the retina. And we're going to work from the picture from top to bottom in the picture. That's the order of flow of information. All right. So towards the top of the image, you're seeing what's called the pigmented layer. It's called that when you look at it under the microscope. It has pigment. It has a color to it. That's not responsible for light. It's going to be that next layer where you're going to find the rods and the cones. It's what we call the photoreceptor cell layer, because that's where you find the photoreceptors. And then they get their names, rods and cones, based off their shape. And you see that in this picture. Rods kind of look long and rod-like, while cones have a pointy triangular tip, like a cone. And so they're going to have to send the information on downward. They're going to have to send the information down towards another group of neurons we call bipolar cells. And they'll synapse with more neurons in another layer called the ganglion cells. And those ganglion cells are sending the information on to cranial nerve 2, to the optic nerve. It's just sending it from neuron to neuron to neuron. We just name these different cells. So that's all that's happening. And then after you get through those ganglion cells, again, you're going to pass it on to the optic nerve. That's all you're seeing in this cartoon is the yellow optic nerve. And yes, there are some blood vessels associated with it too, going in and out through that optic disc in the back of the eye. But we didn't really look inside the eye. We looked at the layers around the eye, forming the eye. What's inside? Well, It's kind of hollow. I told you earlier briefly, it has fluid inside. Turns out when we look inside the eye, it has two rooms in it, two chambers. There's going to be a room in the front, anterior chamber, and there's a room in the back, posterior chamber. How do you know where one room ends and another begins? Is there a border between the anterior chamber and posterior? There is. It's that colorful part of your eye. It's the iris. Again, we'll see pictures again, but we'll see the iris, and we'll see there's going to be a small room, a small chamber in front of the iris. That's going to be the anterior chamber. And we're going to see there's a huge room behind the iris. That's going to be the posterior chamber. And they're going to have liquid inside. And it turns out they're going to have different types of liquid inside. I want you to know the different liquids inside the anterior and posterior chamber. Turns out the anterior chamber has a clear watery liquid, very similar to tears. A clear watery liquid we call aqueous humor. So the anterior chamber, if you were to puncture it, it's going to be a clear watery fluid leaking out. It would be aqueous humor. While the posterior chamber actually has a thick gel-like fluid we call vitreous humor, sometimes just called vitreous fluid. So if you were to kind of poke your eye, it's a little squishy, but it's firm and it bounces back. You're feeling all that vitreous humor in your posterior chamber. Helps to give the eye some of its shape. So when that light comes in, it has to pass through all these fluids. So when that light comes in, it's going through that clear cornea, going through the aqueous humor of the anterior chamber, passing through the pupil of the iris, going through the vitreous humor of the posterior chamber before it finally hits the retina. But it turns out, yet again, there are still more structures. I left out one. Why? Because we're seeing it now. Turns out in your eye, you have a lens, L-E-N-S. Yeah, like a glasses lens or a contact lens. You have a lens in your eye. And it's doing almost the same thing as a pair of glasses or contacts would. It's helping to focus the image, specifically on the retina. So that's what you're seeing on this slide is all the structures light is going to have to pass through. Again, that clear cornea in the front, again, the aqueous humor in the anterior chamber, then the hole in the middle of the iris called the pupil, then the then the lens. It's going to have to pass through the lens, then the vitreous humor of the posterior chamber, and then the retina. And that's what you're seeing on this picture. You're seeing that lens. It's this kind of oval-shaped marble disc-like structure. So now you see all the different structures light has to go through. One more time. Going through the cornea, going through the aqueous humor in the anterior chamber, going through the pupil, the hole in the middle of the iris, passing through the lens, and then through the vitreous humor of the posterior chamber. And then it will hit the retina. But now that we know the structures that light has to go through, we're going to have to explain something else. So we saw when light enters the eye, it's going to have to go through liquid. It's going to have to go through the aqueous humor of the anterior chamber and the vitreous humor of the posterior chamber. This is going to cause a problem. Why? Well, it has to deal with a feature of light, a characteristic of light. Turns out light refracts, meaning it bends. To refract means you bend. And turns out light bends whenever it changes mediums, whenever it changes environments that have different densities, like when you go from air to liquid. This helps to explain what happens what you see when you look inside of something sitting in water, either in your sink or if you're at the pool or in the ocean. If you're in the pool, for example, and you look down, your bottom half of the body in the pool doesn't quite look like the rest of you. Why? You're seeing an image that looks distorted because the light has bent because it's entered the water. So it turns out when light enters your eye, it's going to get a little distorted. Why? Because that light is going to bend. It's going to refract. And what does that mean for us? Well, it means the image that hits your retina is not quite what you're actually looking at. Turns out the image that hits your retina ends up getting inverted and reversed. Meaning it's going to get inverted, flipped upside down, and it's going to get flipped right to left, almost like you're looking in a mirror. And it's up to your brain to correct this image. And you're going to get some help from some structures in the eye, mainly that little marble lens-like structure we saw, the lens. It's going to help you to focus, and it's going to help to accommodate that light as it's entering and bending in the eye. And you're trying to focus that light right where you have your most photoreceptors, right where you have your best image, your fovea centralis. So whenever you're looking around and someone tells you to look at something and you look at it up close, you're focusing it on your fovea centralis. When you're trying to read something and you're squinting, you're trying your best to focus that light on your fovea centralis. That's why your peripheral vision isn't as good as your central vision, because your central vision is you focusing that light on your fovea centralis. And that's all you're seeing when you look at these images. For example, if you were to look at a Christmas tree, even though the Christmas tree is sitting straight up, standing straight up, what's actually hitting your retina is, again, inverted. It's flipped upside down and reversed, flipped from right to left, like looking in a mirror. And your brain is going to have to correct that and put it right back up, right side up, and undo the reverse. And so now you know a little bit of what's happening when light enters the eye. So now we could explain something. Now we could explain something, normal physiology, and when some things aren't working quite right. We could explain the different types of vision. For example, you might have normal vision, meaning you don't need glasses. If you have normal vision, we say you're emmetropic. Meaning when that light enters your eye and it begins to bend, it refracts, your lens is going to do the job of correcting any problems and helping to focus the image directly on the retina. So you can begin the process of interpreting that signal as vision. This is normal. This is what's supposed to happen. Light enters, it bends, and the lens focuses it on the retina. This is normal. This is emmetropic vision. But you might not have normal vision, like myself. Like me, you might have what's called myopia. If you're ever told you have myopia, they're saying you're nearsighted. I'm nearsighted. What does it mean to be nearsighted? Well, it tells you in the name. It means you can't really see something until it's near your face. Meaning you can't really see far away things. It has to be up close. That's nearsightedness. That's myopia. What's the problem here? Well, it has to deal with where you're focusing the light. Turns out in myopia and nearsightedness, you're focusing the image in front of the retina. So you're already focused the image to a pinpoint crystal clear image in front of the retina. So by the time that the light actually hits the retina, it's already back out of focus. You got to think your focus window is really thin, very narrow. So if you focus too soon, what actually hits the retina is going to be already back out of focus. And that's what's happening in myopia. But again, no worries. How do you know? Well, we got a way to fix it, or at least deal with it. It's called glasses. How do glasses work? How do you treat myopia? It's a problem of focusing. You're focusing the image incorrectly. You're focusing it in front of the retina instead of on the retina. So if you want to fix the focus problem, just bring in another lens. That's your glasses. So your glasses are basically helping the natural lens you have in your eyeball. And the type of glasses you have, the type of lens you use matters. Turns out if you want to treat myopia and you just have a plain old lens, you want to use what's called a concave lens. To be concave means you sink in on one side. So you want to use a lens that kind of sinks in on one side. And that's what you're seeing on this picture here. So over to the left, you're seeing what's happening in someone who's nearsighted without treatment. So you're seeing the light come in, it's bending, and the lens is focusing in it. But it's coming to that crystal clear pinpoint focus too soon in front of the retina. So by the time the light actually hits the retina, it's already back out of focus, and that person is seeing things as blurry. But then again, you go and get your concave lens, and now you put that in front of the eye. How does that help? Well, it's a lens, so when light goes through it, it has a different density than air. The light's going to bend. And so it's going to do a refraction first before the light even enters the eye. That's what's happening here over in the image to the right. So light is going to come in, hit the concave lens, bend or refract, then hit your eye, and again, hit the liquid in your eye, bend again, and focus with your lens. But with that extra help, you're now focusing the image where it needs to be. You're now focusing the image directly on the retina. So that's myopia. You're nearsighted. Things are blurry when they're too far because you're focusing in front of the retina. How do you fix it? Put on concave lenses. That's it. But on the other hand, you might have what's called farsightedness. In anatomy, we call farsightedness hyperopia, also sometimes called hypermetropia. But this is basically the opposite of nearsightedness. In farsightedness, it's easier to see things further away, and the closer you get it, the blurrier it's going to get. And now what's the problem? Where are they focusing the image now? Turns out in hyperopia and farsightedness, they're focusing the image behind the retina. It's like they're focusing the image too late. So what's hitting the retina is quite not in focus yet. It's blurry, so they're going to interpret that as blurry vision. So again, if you want to fix this, you're going to need to bend the light before it hits the eye. You're going to need another lens. But in this case, you want to use a convex lens. To be convex means you stick out a little bit, you bow out. And that's what you're seeing on this image. Again, to the left, you're seeing the uncorrected problem. You're seeing farsightedness. You're seeing the pinpoint crystal image focus happens too late. It happens behind the retina. So what's hitting the retina is out of focus. So if you want to focus on the retina and hyperopia and farsightedness, you need to put in a convex lens. So again, the light on the right-hand side, we're following the image. The light will hit the convex lens, refract, aka bend, then hit your eye and the fluid in your eye and bend again. And with your lens, it will now focus on the retina and you'll see the crystal clear image. So that's all that's happening when you go to the doctor and you need some glasses. They're just trying to figure out whether or not you're focusing the image in front or behind the retina, and that would help them to determine what to do next. So I told you, most of being a doctor is just knowing anatomy and what happens naturally, and we could take advantage of it. But again, there are some other problems with vision. You might, on the other hand, have what's called astigmatism. What's going on in astigmatism? If you've ever been told you have astigmatism, what they're really saying is you have an irregular curve to either your cornea and or your lens, meaning they're shaped weird. They have a weird bend. That's all it means. But we know now the curve of things matters. Remember, we saw you could either use an outward bowing convex lens or an inward curving concave lens. Curves matter when it comes to vision because that could bend light in a good or a bad way. So if you have astigmatism, if you have an abnormal or irregular curve to your cornea and or lens, well, that might cause an abnormal refraction of light and cause you to focus the image incorrectly, which you'll interpret as either blurred or distorted vision. So that's all that is. And again, we know some of the pathway of vision already. We know the structures you're going to pass through, all to hit these photoreceptor cells in the retina, all to hit the rods and the cones. But then what happens? How do the rods and the cones send that visual stimuli information to the brain? So we already know the rods and cones, and we already know they're named based off what they look like. Rods look kind of long and cylindrical, like a rod. And cones have a pointy triangular tip like a cone. But why do we use these two cells of all the cells in the retina? Well, it has to deal with how we're going to sense the light, how we're going to capture the light. It's weird, but it's true. We're kind of like plants. Plants capture light with a pigment located in chloroplasts. Turns out we capture light with pigments also, with what we call photopigments, and they're located in these cells. They're located in the rods and cones. So when light hits the retina, it's going to get absorbed by the photopigments in rods and cones, and this is going to trigger a sequence of events that would lead to rods and cones making action potentials. When the rods and cones make action potentials, we call it a receptor potential. It's still just a spark of electricity. So it's kind of similar to smelling and tasting. You're going to take a stimulus and you're going to turn it into an electrical signal. For smelling and taste, we turned the chemical signal originally into an electrical signal. For vision, we're going to turn light energy into electrical energy. That's all that's happening. And we're using photopigments in rods and cones to do it. And this is anatomy. Remember, we name everything. We name the photopigments. We know what photopigments are in rods and cones. Turns out rods have what's called rhodopsin as their photopigment, while cones have different photopigments. This has to deal with their function. Remember, rods really help us in dim light, usually black and white light. When you're looking at this slide, you can read the black words in the white background with your rods, really because it only has one photopigment. So it could really only capture so many different variations of light, and it's rhodopsin. While cones help us with colorful light. We can see all different colors. You can see the red in this slide. If you are looking at other things, you can see different colors with cones because cones have different photopigments that could pick up different forms of light. And it turns out your photopigments absorb light in a cyclical process. That's what you're seeing in this picture here. We're looking in a rod up close. So we're looking at a rod, and the little red discs are the photopigment. You're looking at rhodopsin. And so what happens is when light enters the retina, it's going to hit rhodopsin, and it's going to help to trigger a sequence of these chemical events you're seeing in this cycle. No, you do not need to know this sequence of events. But it just happens in a cyclic pattern where you'll activate or absorb the light, activating the photopigment, and then it'll undergo some processes in terms of what we call bleaching and regeneration. You're basically going to bleach out that photopigment when light hits it. Literally wipe out the color. Then you're going to need some time for it to regenerate until it could absorb some more light. We'll explain what this means in the next couple slides. Remember, stick to the exam review. I told you these photoreceptors are acting in a cyclical pattern where you're going to bleach them and then they're going to have to regenerate. What does that mean? It has to deal with how you adapt to light. When it comes to adapting, there's what we call light adaptation and dark adaptation. Don't be intimidated. They're telling you what's going on in the name. Light adaptation, kind of think that's how you adapt to turning on a light. What happens when you are in a dark room and all of a sudden someone turns on a bright light? That's going to be you undergoing light adaptation. And you also have what's called dark adaptation. Again, this is now the opposite. Now you're going from light into a dark room. You're going to undergo dark adaptation. So what's happening? Let's go through them one by one, starting off with light adaptation. So let's say you're asleep and you wake up in the morning and your mom or dad, brother, sister, whoever comes in and opens your curtain and in screams the sunlight. Oh boy, you're going to have to undergo light adaptation. Turns out as soon as they open those blinds and the light rushes in, you're going to bleach out your photopigments. Literally, you're going to wipe out the coloring. That's called bleaching, kind of like using real bleach. And when it's been bleached, it cannot transmit any more signals. It's almost like the cell is in its absolute refractory period, meaning it can't send another action potential. But no worries, you'll readjust, you'll undergo regeneration, you'll repair the tissue, or the photopigments in this case, in the rods and the cones. and they'll readjust. This is what happens when you turn on a light and you get that bright flash for a moment, for a couple seconds. That's you undergoing bleaching. Everything looks white. Then you regenerate and you readjust. That was you doing light adaptation. It takes seconds. That's what happens when you get that bright flash of light. You were doing light adaptation. You were bleaching out your photopigments, looks white. Then they regenerated, they healed, and they readjusted, and you did light adaptation. But then there's dark adaptation. What happens now when you're sitting in a bright room and they shut off all the lights? Well, you're basically going to do the same thing. You're going to have to readjust. But it turns out dark adaptation takes a while. It takes minutes. It's not like light. You do light adaptation a lot faster because it's more advantageous to see during the daylight. And dark adaptation takes more time. This is why at night, if you were to turn off the lights, everything looks super dark. But if you were to sit in the dark for a while, a couple minutes, maybe an hour, you'll notice it doesn't look as dark as it did before. Or imagine when you first go to sleep and you turn off the lights, everything looks super dark. But when you wake up in the middle of the night, it's almost like an eerie glow, almost like there's some weird night light somewhere. There was no weird night light. That was just you doing dark adaptation. You were adjusting to the amount of light. So that's all that is. And to help you adjust to the amount of light, you're going to have to send signals. Remember, this is all just neurons. When we talk about rods and cones, photoreceptors, this is still nervous tissue. We're talking about neurons. So to help with some adaptation, they're going to have to communicate, and they're going to use neurotransmitters. Turns out, light causes your rod photoreceptors to decrease the release of a specific neurotransmitter called glutamate. Why? Glutamate is an inhibitory neurotransmitter, meaning it stimulates an inhibitory postsynaptic potential in the receptor cell, meaning it's going to keep you from seeing. So when light hits the rod, you actually decrease your production of glutamate, so you're not inhibiting vision and you actually see. So when you close your eyes, you're pumping out lots of glutamate in this dark room to help inhibit the photoreceptors from seeing. Why? Because there's nothing to see. And then when you open your eyes, you decrease glutamate production so that you could go ahead and send the vision signals because there's now light to see. So again, it's a whole sequence of chemical reactions. No, you do not need to know them. That's all this slide was showing you. No, you do not need to know the sequence of chemical reactions. All you got to know is just that you have an inhibitory neurotransmitter called glutamate that's released in darkness because, again, there's nothing to see. And then when you open your eyes and there is light to see, you'll inhibit its secretion from your photoreceptor cells, and you'll be able to send the signal on to the brain to let you interpret light. So let's see that pathway. We know the structures in the eye. You're going to have to send light. But once it hits the retina, where do you send the information next? Well, you know, after the retina, you're going to send the information to the optic nerve. But then where do you go? Well, it's kind of a sequence of other structures similar to tasting. Turns out, if you were to look at the optic nerve at the base of the brain, both your nerves, your left optic nerve for the left eye and your right optic nerve for the right eye, they merge together. And where they merge is called the optic chiasm. And then after they merge, after the optic chiasm, it's like they split again. And we call that the optic tract. One optic tract will go to the right side of the brain. One will go to the left side of the brain. And they'll go as a collection of neurons we call the lateral geniculate nucleus. And that lateral geniculate nucleus is in a place to help you fine-tune sensory information. It's in the thalamus. So after the information hits the rods and cones in the retina, you're going to send it on to the optic nerve, then the optic chiasm, then the optic tract, then the lateral geniculate nucleus of the thalamus so you could fine-tune that sensory information. And then after the lateral geniculate nucleus of the thalamus, you're going to send it on through the optic radiations. Again, just a cluster of neurons. You're going to send it on through the optic radiation to the primary visual area of the occipital lobe. So that's the pathway for vision. And again, that's all you're seeing on this picture. Again, light hits the retina of your eye, send it on to the optic nerve, then it'll merge together at the optic chiasm. That's what you're seeing as that little X in the picture. Then it's like they split again into the optic tracts. Then you'll go to the lateral geniculate nucleus of the thalamus, then the optic radiations, and you'll end at the primary visual area of the occipital lobe. And you'll interpret that signal as vision. So now, like usual, you now know how something works, so now we could complicate it. Now that you know about vision, we could explain a little bit more complicated material. You got to account for the fact that you have two eyeballs, and those eyes are both in the front of your head. Why is this a concern? Well, this is going to give you what's called binocular vision. Bi means two, ocular for your lenses in the eyes, two eyes. You have two eyes for vision that are going to give you two visual fields, meaning two areas to see. There's going to be your nasal visual field or medial, think close to the nose, things as you're going to see close to what you perceive as the center of your vision. And there's going to be your lateral temporal fields, kind of think closer to your ears or where your peripheral vision is. Turns out you're going to integrate both those types of information. And that's what you're seeing on that previous picture. You're showing you multiple colors, some kind of red, some yellow, and some kind of in-between shades of yellow and orange. They're showing you how your brain is breaking up these two visual fields. Turns out all the visual information from the right half of each of these visual fields has to travel to the left side of the brain and vice versa. All the information of the left half of these visual fields actually have to go to the right side of the brain. Remember, we did lateralization of the brain. The right side of your brain controls left side of the body, and left side of the brain controls right side. So that visual information is going to have to cross. And that's what you're seeing in this picture. For example, over to the left, you're seeing left eye, and over to the right, with all the shades of yellow, you're seeing right eye. Well, you got to account for both those visual fields. Each eye has those visual fields. Your left eye has the nasal side and the temporal side, and same thing for right eye. And you're going to have to send some of this information. Turns out the temporal half of the left eye needs to make its way to the right primary visual area in the occipital lobe. And the same and the opposite for the other eye. You're going to have to blend all this information and send it to specific areas, and it crosses over from right to left. No, you do not need to know that for your exam. But that's pretty much the big picture on vision. Now you know. And to finish off this chapter, we finish off with two other special senses, both of which are coming from the same organ in the body. We finish off with the ear. Why? Because the ear is helping us with these last two bits of special senses. Your ear helps you to hear, and it helps with equilibrium. So like usual, we're going to do the exact same things we did for all the other special senses. We're going to break down the structures helping you with hearing and equilibrium. And just real quick, equilibrium is balance. So think balance. And we're going to talk about how they're helping you to do it. And we're going to see all these structures are either in or associated with the ear. And so we're just going to break it down one by one. We're going to spend most of our time talking about hearing. Turns out it's going to be very similar to the other types of senses. You're going to take a signal and you're going to have to turn it into an electrical impulse, an action potential that your neurons could use to communicate with each other. You're going to have to do transduction. But what is the initial signal? What is sound? Well, sound is really vibrations. You're sensing vibrations and you're picking that up as sound. It's traveling in waves and has vibrations. And you're going to have to take these vibrations, these wave signals, and you're going to have to turn them into action potentials that your neurons could transmit. You're going to have to do transduction. In this case, of sound vibrations. And it's going to be structures in the ear that are going to help us to do it. As well as hearing, there are going to be structures in the ear that help us with our balance, with our equilibrium. So we're going to have to look at the ear. And the ear, even though to you the ear seems very tiny, it is pretty small, but in anatomy it's big enough for us to break it up. So when we look at the ear, we're going to look at it in 3 parts. We break it up into an external ear, a middle ear, and an inner ear. So we're going to have to go through all 3 parts when we're looking at the ear. And that's all you're seeing in this picture. You're seeing all the structures we're going to talk about associated with the ear and all the 3 broken up parts. In the small picture to the left, it's showing you the different regions. The external ears in yellow, middle ears, this small area in blue, and inner ears, all this kind of pinkish color, beige color. And then you see the blown up images to the right. You're seeing a lot of the structures we're going to point out as we go through the ear. But we're going to start from the outside. We're going to start from external ear, and we're going to work our way towards the inner ear. That's all we're doing. So when we look at the external ear, we're looking at the outer parts of the ear. And this is anatomy. We're going to name everything. If you were to ask someone to point to their ear, they're going to point to those things sticking out on the sides of your head. It actually has a name. It's not called ear. It's actually only a part of the ear. It's called the auricle. Your auricle is the thing sticking out on the sides of your head. So it has all the wrinkles. It's made up of cartilage. It's very soft and flexible. That is your auricle. Kind of think if you could pierce it with it, when you get an ear piercing, you're piercing the auricle. And there's a part of the auricle people love to pierce the most. It's what people typically call their ear lobe. You'd call that the pinna specifically of the auricle. So if you get an ear piercing, you're piercing your auricle. And when you look at the auricle, it has lots of little wrinkles and little grooves. Why? It's there to collect the sound waves. And then it will funnel the sound waves into your ear canal, where you put your Q-tip. When you're cleaning your ear, you put it into the ear canal. We call the ear canal the external ear canal. Think where you put your Q-tip. And if you were to put your Q-tip in the ear and push, you'll eventually hit a wall. It's your eardrum. In anatomy, we don't say eardrum. We call it the tympanic membrane. All of this is your external ear. It's the auricle on the outside. It's the little tube where you could put your Q-tip, the external auditory canal, and it's your eardrum, that wall, the tympanic membrane. And your outer ear, your external ear, like I mentioned, is mainly capturing the sound. You'll capture it with the auricle, funnel it into the external auditory canal until it hits the eardrum, the tympanic membrane, where it will vibrate and send the vibrations further into the ear. And there's actually something else in the external ear, something else you knew about way back from when we did skin. Way back from when we did sweat glands. Remember, there was a specific type of gland specifically in the ear helping to make cerumen and oil, which will contribute to earwax. Remember, that was ceruminous glands. We're in the ear finally, so yes, we still have to talk about ceruminous glands. There are still ceruminous glands in the ear making cerumen for protection. Remember, cerumen could do things like kill bacteria, keep your eardrums flexible or pliable to help send the sound. Well, you knew about ceruminous glands already. You just probably didn't know it specifically in the outer ear. And then after outer ear, you'll send the signal, the vibrations, further into the middle ear. Why are you sending the vibrations into the middle ear? You want to send it to the tiniest bones in your body. 3 of the tiniest bones in your body are located in the ear. We call them the ossicles. Think back to lab. We looked at ossicles when we did bones. Remember, there's 3 of them. There's the malleus, looks like a mallet, a chicken drumstick. There's the incus, kind of looks like a little flag on a pole. And there's the stapes, looks like a little triangle with a hole in it. You send the vibrations there so they could oscillate. They're called ossicles because they oscillate. They vibrate. And when they vibrate, they themselves will transmit the vibrations further into the ear. This is all about sending those vibrations. You'll collect it with the auricle, funnel it into the external auditory canal, have it vibrate the tympanic membrane, and the tympanic membrane will vibrate these ossicles. And the ossicles I mentioned are going to send the vibrations further into the inner ear. And they'll send it into the inner ear via a little hole called the oval window. And we'll see that in the next pictures. But right now we're just pointing out some anatomy for middle ear. So one thing we're going to see in the middle ear are ossicles, bones. Why? They're there sending the vibrations. But it's not the only thing in the middle ear. Turns out you have another tube. It's called the auditory tube. Lots of names for it, actually. You could call it the auditory tube, the gustation tube, or the pharyngotympanic tube. It's another tube. And it's a tube going from the ear, middle ear, to your nasal pharynx, which is the higher upper part of your throat. Oh, why do I need a tube going from my ears to my throat? Turns out it helps to relieve pressure in the middle ear. When do you need to relieve pressure in the middle ear? When do you use your auditory tube? Well, you might have used it if you've ever been on a plane. If you've ever changed altitudes pretty quickly, like on a plane. You might notice your ears get a little clogged and stuffy. And how do you unclog your ear when you're on that plane or even on a train? Well, you could yawn, you could chew some gum, you could drink something, try to swallow. That's you trying to activate this auditory tube, this eustachian tube, this pharyngotympanic tube to relieve the pressure in the ear. Now you know why it helps to yawn, to pop your ears. You're just trying to connect or open up this auditory tube to allow for the pressure to be relieved by these two connected areas, your ear and your throat. And again, pictures to help explain. Now we're looking at a zoomed in picture of the middle ear. Again, we see a little bit of external ear. I see external auditory canal. You're seeing in this picture, the tympanic membrane is kind of bluish white. The bones, the ossicles, are in yellow. And you see them sitting on top of the tympanic membrane. You see the kind of looks like a chicken drumstick. That's the malleus. Then you see one that looks kind of like a flag on a pole or a hatchet. That's the incus. And then you see the little triangle with the hole in it. You see the stapes. So when the tympanic membrane vibrates, the malleus will vibrate, then the incus, and then the stapes. And the stapes is sitting on top of the oval window, sitting on top of a little hole to allow it to send the vibrations further into the ear. And then down below, down into the right, you see a little tube heading downwards towards the back. It's heading down towards your throat. That's your auditory tube, or your pharyngotympanic tube, or your eustachian tube. So that's middle ear. And I told you, after middle ear, you're sending the sound information into the inner ear. Sometimes referred to as a labyrinth. And when we look at this labyrinth, when we look at the inner ear, it contains some structures in it, some key structures to help with those two major sensations of the ear, hearing and equilibrium. There's something called the cochlea, the semicircular canals. It's not on the slide, but there's also something called the vestibule. These are all in the inner ear. It's going to be the cochlea that's mainly responsible for helping you to interpret the sound signals. And it's going to be the vestibule and semicircular canals helping with that balance or that equilibrium information. And again, that's what you're seeing in these pictures. You're seeing now inner ear. And to kind of help orient you, whenever I look at inner ear structures, to me, the inner ear kind of looks like a big snail, or you're going to see the snail shell. You're going to see the snail's head and almost what looks like little horns or tentacles coming off the top of the snail's head. That's your inner ear structures. So let's break down that snail. I said it looks like a snail shell, snail head, and some weird tentacles on the snail. What are those three major parts? Well, the snail shell, the spiral shell, this orange spiral you're seeing in the cartoon, that's representing the cochlea. Kind of looks like a snail shell. Then there's this little dilated portion, kind of looks like the head of the snail. That's the vestibule. And then coming off the head are these three looping structures. Those are the semicircular canals. So it's going to be this orange little snail shell like spiral responsible for collecting and helping you to interpret sound. And it's going to be the vestibule, kind of the head of the snail, and the three semicircular canals, the three looping structures that are helping you to interpret the equilibrium and balance information. So, but we got to continue that pathway. So sound comes in, vibrates the tympanic membranes, vibrates the ossicles, and I told you it's the stapes that needs to send it into the inner ear. And I told you the stapes could do it because it's sitting on something called the oval window. Well, on the other side of the oval window, it's going to be fluid. It's going to be fluid of the cochlea. And so when the stapes vibrates, and it's sitting on the oval window, it's going to cause the fluid in the cochlea to move. You're basically sending all these vibrations to jiggle some fluid inside of the inner ear. Why? Turns out, well, we're going to name everything. Turns out we named that fluid inside the cochlea. It's called perilymph. And it's going to be located in a specific part of the cochlea called the scala vestibuli. Scala vestibuli is just an area in the cochlea that's going to contain a fluid called perilymph. So again, the stapes is going to vibrate, and it's going to cause this perilymph to vibrate in the scala vestibuli. Then what? Well, this fluid is going to also travel. You're going to create a wave of this moving fluid thanks to the vibrations. And this wave is going to travel to a different part of the cochlea called the scala tympani. Again, we're just pointing out anatomy. We'll talk about what they're all doing later. But again, this is what you're seeing in the picture. Again, I see my weird snail, and we're focusing on the spiral shell. We're focusing on the cochlea. So the stapes will vibrate, and through the oval window will vibrate the perilymph inside of the cochlea, specifically inside of the scala vestibuli, and then they will cause that vibrating fluid to travel into the scala tympani. Just follow the arrows and you'll be able to follow along. While this image is showing a blown up cross section of the cochlea. And this is actually a kind of cartoon depiction of something you're going to see in lab. You're going to look at the cochlea under the microscope, and it's going to look super similar to this cartoon. You're going to see two large pockets separated by little membrane. There's going to be one larger pocket towards the top in your cartoon that's going to represent the scala vestibuli. And towards the bottom is another pocket that's going to represent the scala tympani. And there's this little membrane separating the both of them. We're going to talk about that next. So when the ossicles vibrate, again, I keep mentioning you're going to shake or vibrate some fluid in the scala vestibuli. Turns out it's going to be associated with that little box area I showed you in the previous picture. There's some structures there. Turns out when the fluid vibrates in the scala vestibuli, it's going to vibrate a little sheet of tissue we call the vestibular membrane. And this is just going to transmit the vibrations somewhere else. It's going to transmit the vibrations to a different pocket of fluid called endolymph. And this endolymph is located in the cochlear duct. It's this little circular area between the scala vestibuli and the scala tympani. It's located next to that vestibular membrane. And when that endolymph vibrates, it's going to vibrate a different membrane. It's going to vibrate the basilar membrane. And this is important because this is where the receptor cells are. We call them hair cells of the spiral of Corti, or spiral organ of Corti, or just plain old organ of Corti. When these cells move, they're going to rub against, yet again, another membrane called the tectorial membrane. And when they vibrate or kind of rub against it, they're going to generate, finally, an action potential, which will begin the journey of sending this information, which you will eventually interpret as sound. So that's all that's happening. Sound waves enter and hit or get collected by the auricles, gets funneled into the external auditory canal, hits the tympanic membrane, which will then vibrate. This will again cause the ossicles to vibrate, malleus will vibrate, then the incus, then the stapes, and the stapes will send the vibrations through the oval window to vibrate the perilymph inside of the scala vestibuli. This will then cause vibrations of the vestibular membrane, which will vibrate a different set of fluid or a different pocket of fluid called endolymph located in the cochlear duct. This will then cause the basilar membrane to vibrate, which will cause hair cells in the spiral organ of Corti to move against the tectorial membrane and depolarize and generate a spark of electricity, generate an action potential. And this is a zoomed up image. This is that zoomed up little box we saw in the previous picture. Up top, you see this white coat. That's the tectorial membrane the hair cells are going to brush against. So if you notice, there's something touching that white sheet up top. There's something touching the tectorial membrane. It's all these yellow cells in the picture. The yellow cells in this cartoon are the hair cells. They're just a type of neuron, in this case, to help send sound information. And so you're seeing their fibers leave. The yellow fibers in the cartoon leave and send the information. You know what nerve you're going to use. Oh, remember, what cranial nerve sends hearing information and equilibrium information? Remember, that's cranial nerve 8. That's your vestibulocochlear nerve. So that's all that's happening. The whole point of sending these vibrations were just to shake these little hair cells so they could send that information on as sound. And these vibrations need to eventually leave. So when we talk about vibrating things, it's actually in a circular pattern. There's a reason why the scala vestibuli and the scala tympani are connected. You're just there to allow entrance and escape of the vibrations. And that's what you're seeing on this picture. One more time, collect the sound waves with the auricles. Number 1 in the picture, send them into the external auditory canal. Number 2, hit the tympanic membrane. Number 3, shake the ossicles where your stapes using the oval window. And number 4 will then shake the perilymph in the cochlea, specifically in the scala vestibuli. This will then cause the shaking of that vestibular membrane, which will then shake the endolymph located in the cochlear duct, which will then shake, yet again, the next membrane, the basilar membrane, which will then shake the hair cells, which will then rub against the tectorial membrane and generate a spark of electricity. Then in this picture, you know what happens next. You're going to send that information using cranial nerve 8, using the vestibulocochlear nerve. And that's what you're seeing. That's all these yellow extensions leaving that snail-like shell, leaving the cochlea. You're seeing the cochlear branch of the vestibulocochlear nerve. It's carrying the hearing information. But then where will it go after that? Again, we got to track the pathway. After the vestibulocochlear nerve, well, you're going to have to send it on up. You're going to send it on up to the medulla oblongata, to the cochlear nucleus, singular, nuclei is plural, of the medulla oblongata. You're going to fine-tune some sensory information there, some hearing information. Then again, this is a type of hearing information that's not smell. So after the cochlear nuclei of the medulla oblongata, you got to send it to the thalamus. But to a specific place in the thalamus called the medial geniculate nucleus. Remember, for vision, it was the lateral geniculate nucleus. Well, for hearing, it's the medial geniculate nucleus. And then you'll send it on up to the primary auditory area in the temporal lobe. And again, that's what you're seeing in this picture. Go through all those structures in the ear until you get to cranial nerve 8, vestibulocochlear nerve. Then you'll send it on up to the cochlear nuclei of the medulla oblongata, then to the medial geniculate nucleus of the thalamus, and then on to the primary auditory area in the temporal lobe. That's the pathway of hearing. But don't forget, the ear does one more sensation. It helps with equilibrium. which I mentioned is balance. But turns out you have different types of equilibrium. You have different types of balance. So when we talk about this one, we're going to talk about both of those different variations. What's the difference? Well, what are they and what's the difference? There's something called static equilibrium and something called dynamic equilibrium. What's the difference? Remember, don't be intimidated by the names. Their names are actually giving you a hint. First is static equilibrium. Static, as in you're pretty much staying still. Static equilibrium is relating your balance. It is relating your body's position relative to gravity. When you're talking about balance, this is when your body's constantly comparing your position to where gravity is pulling it towards the earth. That's all it is. So as you're sitting in your chair, you're feeling the pull of gravity to keep you in the chair on the ground. You're able to feel your relative position relative to that force of gravity as you're sitting there, thanks to your static equilibrium. This is different from dynamic equilibrium, which is your body's ability to sense its position in response to a sudden change in movement. Kind of think when you're sitting in the car and the car pulls off and you feel that drag, your body needs to sense its position change when you're moving. That's dynamic equilibrium. And so it's going to be the vestibule and the semicircular canals and structures within them that are going to help you to do it. So what are they? We call all the structures helping with equilibrium your vestibular apparatus. And there are going to be specific structures within your vestibular apparatus. They're going to be something called the saccule and the utricle and your semicircular canals. And we call the saccule and the utricle otolithic organs because they have otoliths. Oh, what is an otolith? It's just a calcium-based crystal. It is literally a crystal. You have tiny microscopic crystals floating around inside of your ear, specifically in the vestibule of the ear. And we'll see these otoliths, these calcium-based crystals, are going to play a role with other structures to help us sense our different types of equilibrium, like static equilibrium. Turns out when we look inside, we'll see these pictures. I'm just letting you know they exist right now. But when we look at the saccule and the utricle, they both contain what's called macula. Remember, macula, like in the eye, is just a collection of cells. The macula are where you're going to have your receptor cells to sense specifically static equilibrium, for example. And just like with hearing, once we collect this equilibrium information, we're going to have to send it on. Those otoliths and the otolithic organs, the saccule and the utricle, they have what's called an otholytic membrane sitting on top of the macula, sitting on top of these cells. So when your head moves, thanks to gravity, there are also hair cells here. These hair cells are going to move and they're going to generate action potentials as well. When sound comes in, hair cells make action potentials. And when your head moves or body moves in relation to gravity or sudden movements, the hair cells will still move and you'll collect the information based off the source of movement. And you'll use cranial nerve 2 to send the information. But when you're sending equilibrium information, you're going to use specifically the vestibular branch of cranial nerve 8. So that's all you're seeing in this picture. Now we're looking at the vestibule, looking at that head-like portion of the snail. That's where you're going to find the saccule and the utricle. That's your vestibular apparatus, is the vestibule I mentioned. When you look inside, you're going to find all your neurons, which are still hair cells. And in this picture, you're seeing the otholytic membrane sitting on top, kind of like that tentorial membrane we saw in the organ of Corti. It's just that in this case, this will move in relation to head movements, and it will cause the hair cells to move, which will then cause an action potential to get generated, and you'll send the information on using cranial nerve 8. And this one is showing a blown up image where you're seeing the small crystals inside. You're seeing the otolith. So when your head moves, these crystals move, which causes the hairs to move and generate an action potential. So that's the saccule and the utricle. That's your vestibule. That's that head-like portion of our snail when we're looking at the inner ear. But then there's the semicircular canals, those three looping structures. They're helping you to sense dynamic equilibrium. They're helping to sense your body's position in response to sudden movements. And when we look at pictures of the semicircular canals, they look kind of crazy. They're all going in different directions. It's for a reason. Remember, everything looks a certain way because it has a certain function. When you're looking at the three semicircles, the semicircular canals, you're looking at tiny ducts. There's liquid inside. And they're going at different directions to help with movements. Remember, dynamic equilibrium is your body's position in response to movements. Like when you're in a car, I mentioned. When you're in a car, it will accelerate and you will notice the difference in the acceleration in your body's position. And you'll notice your body's position change when the car decelerates. Well, your semicircular canals are going in different directions to allow you to kind of pick up and sense what we call rotational acceleration and deceleration when you're moving, basically. And again, it's going to be cells sensing this movement. There's going to be cells sensing this sense, these stimuli. And again, it's still hair cells because we're still in the ear. But now we're in the semicircular canal, so things going to have a different name. Turns out each of those tubes, each of those three looping semicircular canals has a little entrance we call the ampulla. And in the ampulla, you're going to have a collection of hair cells we call the crista. So when your head moves, the liquid inside the semicircular canals is going to move, and that liquid is called endolymph, and it will cause the hair cells in the crista within the ampulla of the semicircular canals to move. And when those hair cells move, like all the other hair cells, they will generate an action potential that will send the signal. And we already know we're going to send the signal using the vestibular branch of the vestibulocochlear nerve. So that's how you're going to pick up both of those types of equilibrium. And again, that's what you're seeing in the picture. Now we're at the beginning of each semicircular canal. We're at the ampulla. And then there's going to be a collection of hair cells. We call that the crista. And so when you turn your head or have any sudden movements, liquid, the endolymph inside of these tubes is going to move and cause the hair cells to move, and they'll send the signal. That's all you saw. When this lady in this picture turned her head, the liquid moved, which shifted the hair cells, and they generated a spark of electricity to let her notice the change in her body's position. But where do you send the information after the vestibulocochlear nerve? Like always, we got to kick it on up to the higher parts of the brain. Well, after the vestibulocochlear nerve, for equilibrium, you're going to send it to the vestibular nuclei of the medulla oblongata. Then after the vestibular nuclei, this is still sensory information that is not smelling, you still have to send it to the thalamus. Specifically to the ventral posterior nucleus of the thalamus. And then after the ventral posterior nucleus of the thalamus, you'll send that information to the vestibular area of the cortex. So again, vestibulocochlear nerve, then vestibular nuclei, then ventral posterior nucleus of the thalamus, and then your vestibular area. And that's how you're going to interpret equilibrium information. Again, lots of summary slides. I know these are lots of weird structures, so there's lots of summary slides to help you through it. That's all you're seeing on these images on table 17.2. It's just going through the ear from outermost to innermost. So on this first table, you're seeing outer ear, and it's reminding you the outer ear has the auricle, the external auditory canal, and the tympanic membrane, and it's reminding you of their function. Then the next slide will go on to the middle ear, reminding you that's where you're going to find all the tiny bones, the ossicles, and the auditory tube, sometimes called the eustachian tube, sometimes called the pharyngeal tympanic tube. And again, it's telling you their functions. And then it goes into the inner ear, again, breaking down structures here. Remember, inner ear has a lot of structures because it does multiple senses. But overall, think of it as a snail with a shell, spiral shell, cochlea. The head, which contains the utricle and the saccule, That's the vestibule. And then there's the 3 looping ducts. We call them the semicircular canals. And it reminds you of their functions as well. So you know all about your special senses. So to finish off this chapter, we just hit a little bit of development. And then like usual, we talk about what happens when you get older. But first up, we're going to mention just a little bit of development. We're really going to focus on the development of the eyes and the ears. And we're going to start off first with the eyes. Well, it turns out when you're developing in the womb as a fetus, your eyes per se won't begin to develop until you're about 22 days after conception. 22 days after fertilization, what's considered you is going to begin to make eyeballs around 22 days. And you got to think, eyeballs we saw had multiple layers, but the layer that mattered was the retina. And we know the retina really contains a lot of sensory receptor cells, the photoreceptor cells, which are just neurons. So think nervous tissue. Think back to when you were just a flat pancake. At 22 days post-conception, you're 22 days into gestation, you're pretty much still that flat pancake of endoderm, ectoderm, and mesoderm. And you remember which layer did nervous tissue come from? Ectoderm. So your eyeballs, which has a lot of sensory material, nervous tissue in it, will begin to develop from the ectoderm. And then it will form what we call optic grooves, which will become optic vesicles, and you'll begin to progress. And we just name different structures as you're progressing. Remember, trust the exam review. I will only ask you what's in the exam review. This picture is just showing you a lot of those structures that were in bold, helping with your development, highlighting all the structures you go through in your development. I will not ask this. Remember, trust the exam review. So again, you begin to make the eyeballs after 22 days of gestation. And you're making it from ectoderm. And it will go through a series of progression into other structures until you begin to form the eyes. That's all these slides are going through. I'm not going into it in detail because, again, I'm not going to ask you. And again, like usual, there are pictures. Trust the exam review. So to finish off this chapter, we just talk about what happens when you get older. You know already it's not going to be good. And that happens for all your senses. Remember, we've seen now all your senses are thanks to cells. And you know by now, as you get old, your cells get old and they die. Or as they get old, they don't work as well as they used to. Same thing for all these cells, for all these senses, like smelling and taste. But turns out these cells could last a pretty long while. Turns out you won't begin to notice any deterioration in smelling and taste until you're around age 50. That's when you might notice a gradual change in taste and smell because you're gradually beginning to lose these receptor cells, the olfactory receptor cells for smelling and the gustatory receptor cells for taste. And you're not going to have enough basal cell activity to help keep up with the replenishment. So you're going to see a decline over time. Why? Because your cells get old like you. And you'll see changes in your vision as well. What structures are going to change to allow for changes in vision as we get old? Turns out it's going to be the lens. You saw when we talked about vision, the lens is very important for focusing the light so we can see clearly. Well, it turns out as you get older, your lens loses its elasticity. Your lens could change its shape pretty easily when you're young because it's flexible. And it needs to do that. Turns out when you look at something up close, your lens kind of relaxes and gets kind of plump and round, almost like a round marble. But when you're looking at something far away, your lens stretches and looks flatter like a little disc, a lens. But when you get older, its elasticity, its flexibility decreases. So its ability to change its size between looking at something up close and far away is going to get harder. And it's going to get really hard to kind of form that round ball when you're looking at things up close. So this is why grandma and grandpa have a hard time looking at things up close. That's why when you give grandma or grandpa that birthday card, they look at it, then they hold the birthday card far away, and then they hold it up close, then they hold it far away. They're struggling to see it clearly, because you would think, oh, can I just hold it up close? No, because they're having a hard time adjusting the shape of their lens because it's not as flexible. So up close things are going to be blurry. But we're not going to call it farsightedness. We're not going to call it hyperopia, because the problem is not because they're focusing the image behind the retina. It's because the lens isn't even changing shape. We call that presbyopia. Okay. Starts to happen usually around age 40. So as early as age 40, you could start to need your reading glasses. Now you know why grandma and grandpa need reading glasses. Their lens have lost their elasticity, so it's hard to see things that are up close. Keep going. Not only that, the muscles in the iris, remember your iris has muscles to help change the shape of your pupil. Turns out the muscles of the iris get weaker. And when they get weaker, they don't work as fast as they used to. So grandma and grandpa have a hard time adjusting to light changes because their pupils aren't dilating and constricting as fast as they used to. Remember, knowing anatomy helps to explain a lot. Keep going. When you get older, you increase your risk for certain diseases as well. Diseases of the eye especially. You could get what's called macular degeneration, where that macula lutea, where you have a lot of photopigments for nice vision, and the fovea centralis, well, that could deteriorate. We call that macular degenerative disease. You'll have problems with vision. Or you could have a retinal detachment. If you've ever been told you have a detached retina, it's what you think. You saw the eyeball has layers to it. And that inner layer, the retina, it could literally detach itself from the other layers. It could literally peel off. What tends to do it? What tends to cause a detached retina? Things like high blood pressure, all that blood coursing through the choroid. That could help to detach the retina. What else? Or you could get glaucoma. Yeah. Some people always get tested for glaucoma when they go to the eye doctor. What is glaucoma? Glaucoma is just increased pressure in the eye. That's why sometimes when you go again to the eye doctor, you sometimes get a little puff of air in your eye. That puff is them checking the pressure in your eye. They're checking to make sure you don't have glaucoma. It's just increased pressure. That's bad because the increase